All right, now you can really hear my dog, I think. I'm gonna share my screen. Oh, dog, really? Hey. Okay, so this is where we left off, thinking about how bacteria are so good at picking up pieces of DNA through one method or another. This is the one that we're thinking about with regard to plasmid transfer, or at least one strand of a plasmid getting transferred. And you can see here that there are different phenotypes. If the bacteria can gain plasmids, you can see different things that this would allow them to do. And again, if it's suitable for their habitat, if it's adaptive and it helps them better cope in their habitat, they will maintain these plasmids. However, if they get a plasmid that is really of no use, they can purge it. And so it's not like they always have to keep every plasmid they get, because over their lifespan, they can get an appreciable amount, you would imagine. And so it would get a little too cumbersome. And so bacteria are actually quite good at purging, getting rid of unnecessary DNA, whether it be a plasmid or stretches of their gene segments on their actual circular chromosome. So again, if you have that plasmid conferring a selective advantage, then it would be to the benefit of the microbe to go ahead and hang on to that and then pass it on when they go through cell division and create progeny cells to pass it into the progeny cells. And so it can be pretty effective, pretty easy for these bacteria to do this. And like I said the other day, you can do it in the lab setting if you understand your microbe and how to manipulate them in order to get them to take up a plasmid. You grow them up usually in a broth culture and introduce the plasmids. So you can get these plasmids transferring. You can get these plasmids transferring throughout a population, and from our perspective, that can be a bad thing when you think about antibiotic resistance, because many of the microbes that are antibiotic resistant, when you look at them and say, okay, where are these antibiotic resistance genes? A lot of them are carried on plasmids, and so that's a big problem. And we talked about yesterday how plasmids can be transferred between and among really distantly related organisms, including organisms in different domains. And so when that first came out, again, it was shocking because we didn't think that this would be something that could cross domains, so to speak, this behavior, and yet we see that it's more prevalent than we first initially thought it would be. And so we can see that there are bacteria and fungi that can exchange DNA. Bacteria and plants can do it. And so there's some really crafty bacterial pathogens of plants that the way they create disease in the plant is through this process of putting one of their plasmids and getting it pasted into the plant chromosome, which is really quite amazing and fascinating. And then that allows the bacteria to essentially gain access to nutrients that it couldn't get otherwise. And of course, the bacteria has a nice home inside the plant. And so it's, from the bacteria's perspective, it's a pretty great adaptive trait to have.
Would you say this is the most common form of horizontal gene transfer?
The conjugation, it's been the most heavily studied, I'll put it that way. We have a great interest in this, primarily because from the human perspective, we know that antibiotic resistance, that's the main way that that trait gets spread. And so we studied it the most, and so that might be biasing how we are seeing this happen and kind of tallying up what's the most common way. So I would be cautious in saying plasmid transfer is the most common. I think in natural settings, you also have to factor in that when a microbe's living, like say, for example, in the soil, you have millions of other bacteria nearby that could be lysing open, releasing DNA. And so transformation may actually be really prevalent. And it's just, we haven't studied it very much. And unfortunately, it's hard to track that easily. So there's a long answer to your question. So, all right. Another table from a different source, looking at, again, what I like about this is they're getting at the idea that you can see transfer between distantly related microbes. So if you look at the middle column, lineages involved, you can see how there's transfer from, bacteria to yeast, bacteria to fungi, and then they get into eukaryote transfer. All right, from fungi or eukaryotic, even though we consider them microbes, they're eukaryotic organisms. And so you can see that, the more that we study this, the more we're finding these amazing examples of plasmid transfer among really distantly related organisms. And so seems to be a common theme. So it lends you to start thinking about this idea of the phylogenetic tree might not be quite what we originally drew, so to speak, and how it's, kind of this tree that goes straight up from the last universal common ancestor like we normally see the drawings of a phylogenetic tree. Here, this artist drew the example of, we need to think about this idea of horizontal gene transfer and how it really can be playing a powerful force in the process of evolution and speciation. And so this idea of, let me get my laser pointer, how you see this crossing amongst the domains horizontally. So they're trying to get at that point that through horizontal gene transfer, that is also playing a role in speciation and life on Earth. And so I think, I don't know if anyone's taken evolution here at Northern, if they get into this at all. I don't know. Has anyone had evolution? Does this sound like something they might have covered in that class? Do you remember it?
Yeah, that might
just a little bit, but not too much.
Okay. Yeah, it is something that is, I guess, more commonly talked about if you're an evolutionary microbiologist. This is kind of like, well, yeah, of course, but I don't know how much this has gone into the world of people who are more macro evolutionary people. So, and then when I go to conferences, I usually either go to a plant conference or a microbiology conference, and we see this is kind of an accepted idea in those arenas, but I don't know how widely accepted it's been, because this is a fairly new idea. And so again, this bottom picture, if you compare that to the top of the phylogenetic tree comparison, it's been, most scientists will say that this idea of endosymbiosis is quite plausible, and that might have actually happened hundreds of millions of years ago. But with more study, we're now saying, horizontal gene transfer really can be playing a much more major role. And we now know that it can go, between organisms from very different domains. So I think it's an exciting time to be thinking about these ideas and looking at relatedness, and then, how did these organisms gain these special traits, and the whole speciation process. It's a lot messier than we first thought, which, again, whenever you're in science, right, things get more and more complex the deeper you study it. That's the fun of science, in my opinion. So, all right. We have, this was taken from a published study. I love this also because it's really showing in a very visually graphic way that, looking at the phylogenetic tree of life, you see that the bacteria domain is really dominating, right? You see the eukaryotes shooting off here, the archaea, but really trying to drive home the point that microbes, bacteria, were here first, before us, long before us. And so they've had, a couple billion extra years of time to exist and adapt and go through speciation and evolution. And so this is some of the results on this that they got when they published the study is through the idea of, they didn't necessarily isolate every single one of these microbes and study them in the lab. It's looking at DNA samples and sequencing the DNA from, raw samples in the environment. So they would take water samples or soil samples, and then rather than isolate the microbes themselves onto Petri plates, they would take the DNA and extract it and do what's called metagenomics, and we'll talk about that in the not too distant future here. So really, bacteria rule when it comes to life on Earth here. There's more bacteria by far than any other living organism. And when you think about all the microbes in the oceans as well as on the terrestrial surface. So, and then in the deep subsurface, we're realizing microbes hang out deep, deep into the Earth's crust and are totally happy there. So we'll get into some of those ideas as well. One other thing I need you to appreciate, which often is not emphasized probably anywhere else than in a micro class, is that prokaryotic organisms, they are what we say genetically haploid. They only have one copy of each gene, whereas you and I, we have two copies, right? We got one copy for eye color that came from our mom, one that came from our dad. All right, so prokaryotes they're in a little bit more precarious position, if you want to think of it that way, because of this, they're genetically haploid. And so if that gene malfunctions or gets a mutation, that could be disastrous for the microbe. And so microbes have this ability to go through this process called gene duplication, which as they are replicating their DNA, they can go through and make like an extra copy of a gene. And I still don't know all the nuances of that process. I have not gotten into the study of that in any deep way, but have read enough where it's convinced me that, yeah, this is actually something that can happen under natural situations. And so it's a way for them to kind of have an extra gene in their back pocket, so to speak, that they can pull out and use if need be. And then, of course, the horizontal gene transfer too, right? That can also really help them get access to critical genes that would really help them. And so again, too, just to refresh your memory, when we say the word mutation, that's just a change in the nucleotide sequence of the chromosome, all right? In common culture, the word mutation tends to have a negative connotation. And, even people will throw insults at each other and say, oh, you're such a mutant, and that's taken as an insult. And so I just want you to appreciate that all it really means is it's a change. It could be a change for the worse, certainly. It could be a change for the better. And then some mutations are null. They don't really affect anything critical on that chromosome. So it is something that is, you got to be a little bit more neutral about it when you're in a science class. It's not necessarily a bad thing. In fact, it can be really good for especially bacteria. So mutations can happen in many different ways. And in the lab, for example, you can force a mutation event by zapping microbes with ultraviolet light. If you give it in sublethal doses, you can certainly cause mutations on the chromosome, because ultraviolet light, that's the way it impacts our cells, is it tends to damage our DNA. And you can also have mistakes being made during the replication process as that microbial cell is getting ready to go through cell reproduction. And so, and then other factors I put in italics, because horizontal gene transfer is also, you can think of it as a mutation in the sense that the bacteria is getting new genes that it never had before. And so you have to factor in horizontal gene transfer as well as mutations, especially when it comes to microbes, the bacteria in particular, because that can profoundly impact how they adapt, survive, and then the speciation process as well. Another thing to appreciate, microbes, we talk about how microbes can gain DNA, but they can lose DNA, and they can do it on purpose, so to speak. And so gene loss is something that is well documented in the microbial world. This is something where it makes sense, especially if you are an obligate parasite or an obligate symbiont, where you have to be with that host or you die. And so there are many examples of that have been studied, where they can see that if you try to have this parasite or pathogen exist on its own away from the host, it doesn't make it. And then when they look at the DNA and say, why isn't it living? Why isn't it surviving? It's because they've purged so much of their DNA that is critical for life being lived alone and separate from a host. They purged it all because they don't need it. They're so tightly and intimately associated with their host over time that relationship became so intimate and long-term that they purge the DNA that they don't need anymore because they can use their host for that purpose. And so gene loss is actually a quite good strategy for certain situations. And so keep that in mind too as we're thinking about the microbes and how they deal with gain or loss of genes and bits of chromosomes and plasmids. So adaptive mutations improve fitness of an organism, increasing its survival. All right, so you have to think about, again, really looking at the microbe's current environment. That mutation might be adaptive under one habitat. You pick that microbe up and put it into a different habitat, and it could be disastrous. That whatever that mutation that it recently got, it could be really bad if you move it into a different habitat, or if the microbe's habitat changes greatly. Even thinking about climate change, for example, that can have an impact on shifting the environment where now that original adaptive mutation no longer is going to work. Okay, so when you think about it in the microbial world, you can get speciation happening from this process of natural selection that you've heard about if you've taken a more straight, normal evolutionary class. But you also have to think about the horizontal gene flow idea as well. So both of those you have to keep in your head when you're thinking about how are microbes adapting, and then the process of evolution and speciation. And so again, going back to that quote, whenever you're studying the material in this class and onward, and once you get out of this class, thinking about how, through thinking about ideas and what's happening in whatever ecosystem you're studying, keeping that lens of evolution, looking through the glasses or the lens of evolution and the evolutionary process, I think can really help you better understand what might be happening in that particular ecosystem. So I'll keep trying to remind you of that as we work through the content in this class, because I think once you start thinking that way, the material starts to make more sense. Okay, in EduCat I did post this next slide set, and it's the ecological physiology slide set. Whoops, I need to, hang on. I'm going to share my screen. So hopefully you can see the first screen, ecological physiology. So those of you who have taken Gen Micro with me, you know that we did cover physiology in that class quite heavily. I'm not going to do it to that extent in this class. However, I do have to give you a little bit of physiology so that you can relate that information to the particular ecosystem that you're studying and thinking about what are the microbes doing there. And this will also help you for your field guide assignment, where I do want you to think a little bit about that as you go to your field site and you're you know, going to start looking into the published literature, like what are these microbes doing physiologically in these habitats, and how might that be altering the habitat in any way. So when you think about microbes, first of all, when you took Gen Micro, if you took that class, you looked under the microscope and saw different shapes of bacteria, and you were most familiar probably with the so-called coccus shape, spherical shape, and then this rod shape. Those are very, very common in the types of microbes that we work with. But you need to appreciate that there are many other types of shapes and designs that you can see. Spirillum is another, and here are the actual pictures under the microscope on the far right. So spirillum shape, there's actually some key human pathogens that are spiral and spirochete shaped. And then there's also some that look very, they're not symmetrical. They have distinct parts to their body, so to speak. So here you see a stalk that allows them to attach. So a lot of microbes that are in running water systems, for example, rivers and streams, they tend to have, many of them, like they'll have a stalk so that they can attach to rock surfaces so that the running water, the current, can't rip them away from their habitat. And so they'll use that for attachment. And then there's plenty of filamentous bacteria also, which, you know, when you look at this last image, some of you who are familiar with fungi, you might say, well, that kind of looks like a fungi, but they've labeled it a bacteria. There are plenty of filamentous bacteria. In fact, one of the major groups of bacteria that I study are called the Streptomyces bacteria. And Streptomyces were once thought to be fungi when they were first discovered because they have these petite, and we actually use the word hyphae, which we typically say about fungi. We say, look at the bacterial hyphae, all right, the long hair-like cell design. Streptomyces do have that hyphal growth, but their hyphae are much more petite, slender, smaller in diameter compared to fungal hyphae. Fungal hyphae look huge in comparison to the Streptomyces hyphae, for example. So in natural habitats, you will see lots of differences in design of the bacterial body. The microbes that hang out with us, with humans that are in and on us on a regular basis, they tend to be more coccus and rod shaped, although I've done some teeth scrapings where there's bacteria, there's relatives to the Streptomyces that are well adapted to our mouth. And so that's always kind of like, whoa, I got hyphae in my mouth. It's kind of an interesting thought. One other thing you should remember too is that when we get down into the micro world, we see the surface to volume ratio go up. All right, so the smaller the cell, if you do the math, which I'm not going to ask you to do, but here it is, to convince you that the surface to volume ratio increases as you decrease the cell size. And they do a nice comparison here to a bigger cell, and you can see the difference. And that is actually, they think, a good thing for microbes. When you think about their design, being small gives you some advantages because you don't have those internal organelles. You don't have those. Instead, your cell membrane actually is incredibly important when you're a microbe, because that's where the microbes put a lot of their proteins that are involved in really key physiological reactions. So they don't have an internal mitochondria, right? They don't. They're bacteria, and they just don't have that. So all the functions that the mitochondria, the respiration functions, that gets put into the membrane instead, the cell membrane. So for a bacteria, the cell membrane is not just a barrier and a selective membrane. It is also a key spot for physiological reactions. And so that surface to volume ratio is pretty high when you get small like that. And it helps with them to be better able to function because of their internal design lacking organelles, and then the speed at which microbes need to get things across their membrane. This surface to volume ratio being very high is a good thing. One other thing, though, that you need to think about is that bacteria are pretty good at altering their physical size, and this is often in response to what's happening in their environment. So when you grow bacteria in the lab and you give them all the conditions that they could absolutely want in order to be fully functioning and really cranking out their physiology, they'll reach a certain size. But then if you take those bacteria and plop them into some broth, let's say, where you start starving them of key nutrients, you can often see the bacterial size will decrease. They'll get smaller. And so I tend to talk about feast or famine conditions. If you can imagine the bacteria under feast conditions where they're getting all their needs met, they can ramp up to their full potential in terms of their size. Famine conditions, on the other hand, they can be very flexible and shrink down, and that helps them survive tough times. And so, obviously we don't do that, although, if we don't eat proper nutrition, we don't grow to our full potential either physically. But bacteria have this flexibility, in a matter of hours where they can shift their actual size. And so that's a coping strategy under famine conditions to survive through it, and then hopefully they'll enter back into a feast condition and can go back to their normal size. This is just getting you to think about the different sizes of microbes and the idea that we do see a lot of range in the overall size of microbes that have been studied. So on the very bottom, Mycoplasma pneumoniae, that's the one that causes walking pneumonia in humans. And you can see that it's tiny, 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 right? And the cell volume also really, really small. And then you go all the way up to the top, Thiomargarita. I love that word. Thiomargarita namibiensis, sorry. That was discovered in Namibia, Africa. And I think Thiomargarita means sulfur pearl. Thio is sulfur, and I never knew that margarita meant pearl. I thought it, margarita, it's an alcoholic drink. What are you talking about? But when you look up the word, it means pearl. So sulfur pearl of Namibia is how they named this particular microbe. And it's huge. You can see it. If you got really good eyesight, you might even be able to see it with a naked eye. So that's, I might have a picture of it here. Oh yeah, right here. So it's very big. And you don't have to memorize all this. I'm just, if you're curious. And so here you can see it's kind of like pearls on, a pearl necklace. That's why they named it that. And so massive. And again, if you have good eyesight, you might even be able to pick it out with the naked eye. Here's another one that's really big. It's kind of fun to think about. E. Pulopicium fischelsoni. So any of you who are familiar with fish, they actually discovered it in the guts of sturgeon fish and found that it was huge, or surgeonfish, I should say. And so again, 0.6 millimeters, that's big, all right? We usually don't get any bigger than 0.45. And interesting design, it has a lot of membranous material that goes internal. It kind of infolds into the interior of the cell, and that helps it increase its surface area because it's so big. So look at this difference. So here's the bacteria, E. pulopicium. There's E. coli, that little black dot. And here's paramecium, which I'm hoping you have had a chance to see at some point in your life under the microscope. They're so fun to look at with all their crazy movements. And so paramecium, those are eukaryotic organisms. So you might have learned in BI 111, they might have said, prokaryotic organisms, they're always smaller than eukaryotic organisms. Well, again, you can't say always or never in science. Here's an exception where you have this giant bacteria. So there's more out there, I'm sure. They're there lurking. We just haven't discovered them yet. Here is a pin. I zoomed into the head of a pin, and you can see E. pulopicium on that. And so just to compare the size difference, most of you are familiar with a pinhead. So fun things to think about with that. Okay, so when we start looking at the microbes and getting down into their DNA, how big is their chromosome, how many genes do they have on their chromosome. It's easy to do that now with the technology that's available, and the sequencing of microbes has gotten much cheaper over time. And so this is something that's pretty easy to do. And so in the microbial world, we use the term open reading frame. And so that's jargon that you'll see if you ever have bacterial DNA sequenced. They'll talk about open reading frames. And so that is analogous to a gene. And when you start mapping out the genes in microbes, we'll see that there are different overall genome sizes in terms of, I guess, the number of genes in total is what I'm thinking about here. And so, and then when you also factor in horizontal gene transfer, so now when you start thinking about speciation, especially down in the microbial world, it's kind of this disconnect, because you can say, you know, these two organisms are really closely related, and yet the way they behave can be really different. So their phenotype, their outward behavior of the expression of those genes, and then what's actually on their chromosome or in their plasmids can be different. And it's mainly, we think, due to so much of the horizontal gene transfer that is going on in the microbial world. And so just because you see the two organisms behaving the same way doesn't necessarily mean they're closely related. And that's kind of a different thought process compared to maybe what you've been learning when you look at macro evolution of, you know, the big fuzzy animals. And so here we're looking at prokaryotic genome sizes, and they give it in megabase pairs and also in the open reading frames. And so you can see that there are different amounts of genes showing up on these, in these different microbes, even though they're closely related. So let's go down to E. coli. Here you see E. coli K12, E. coli O157H7. Okay, they're both E. coli, all right, Escherichia coli, but they're different strains. So the K12 strain and the O157H7 strain, and you can see this huge difference in their open reading frames, okay? And you may, that O157H7 may sound vaguely familiar, and it should sound familiar to you, to those of you who've had Gen Micro or Med Micro, because that's the human pathogen that is often found in contaminated beef, and, well, not just beef, but other meat sources, and can cause death in humans if you get a large dose of it when you're eating improperly cooked and handled meat.
The ORF is what makes them different.
I didn't hear that. Could you repeat, please? Oh, you're breaking up. Maybe you could chat.
ORF is what makes them different. Everything else is the same.
So the open reading frames, yes. So essentially, I had a really good, I'll see if I can try to find it. I had a really good reading that looked at what's the difference here? Why did E. coli O157 have this huge shift in the number of genes? And they were able to finally solve that mystery, where they could show that this particular E. coli gained large chunks of DNA through horizontal gene transfer. And then on some of those bits of DNA, it allowed them to become pathogenic. And it was a stepwise thing. It didn't happen all in one fell swoop. It was this, at least what they published on this paper, it was this stepwise progression. And so there's like intermediate strains, and then you have the fully formed O157H7 that is really potent at causing disease. And so yes, it's just they've largely, these microbes are acquiring DNA largely through horizontal gene transfer, and that's what's causing these big shifts in the open reading frame counts. Where is the other one I wanted you? Oh, here, Carsonella rudii in the middle of the chart here. And you can see this tiny number, 182, and it's an obligate endosymbiont, all right? So obligate, it has to be with its host. Endosymbiont means that it's within, which is an insect. A psyllid is an insect that, in my opinion, is nasty because it attacks plants and feeds off the sugar of the plant. And so, but it's become so, through coevolutionary processes, and it has become so intertwined with its host, it's purged a lot of its DNA, right? So here's an excellent example of a comparison of a microbe who has purged DNA and only kept, bare minimum. So kind of fun to think about, and then, wonder about, well, what happened to cause these shifts? And it's, some they have figured out, and others they still don't know the full answer to that. But here's more of that table. So down here, you're also looking at archaea, the domain archaea, which are some really fun, bizarre microbes to think about. But you can see big differences in the open reading frames, the numbers here, comparisons. Again, you see nanoarchaeum, equitans, that makes you think about horses. I'm not quite sure why it was named that, because it does, well, maybe an intracellular parasite, maybe it does infect horses. But they also are finding it in hot springs, so that kind of is weird. I'll have to look into that one. Why would they name it equitans? Equi is referring to horses. Okay. Again, here's Pseudomonas up here, Pseudomonas fluorescens and Pseudomonas putida, so the same genus, different species. And that's not too far of a difference between the open reading frames, all right? So it's kind of all over the map here with regard to, closely related microbes can have similar or different. It's just, it can get kind of confusing if you're unaware of some of the traits microbes have in terms of their ability to get horizontal gene transfer. So I just took a few from off that chart and put them here to show you, some nice comparisons where, look, these are two really closely related strains, and yet you can see a sizable shift here in the open reading frames. So, again, if you don't understand how microbes can operate with the horizontal gene transfer, this could be really confusing. And again, going back to the E. coli comparison. Okay, so keep that in mind, that you can often see closely related organisms behaving very differently. And then you go in and start looking at their open reading frames and you find out, it's because they have a big discrepancy between their open reading frame counts. This chart is looking at kind of grouping what these genes are doing for the microbes. So obviously you have to have a way to store information and process information if you're going to stay alive. And so this is Pseudomonas putida that they're using as an example. And so this is kind of nice, just getting you to think about, well, what are they using in general? What are they using these genes for? And they've grouped it, in these different categories. So, the bolded things here, I would like you to think about these ideas and understand that there's some major categories of how these, genes need to function for the microbe to stay alive and be able to reproduce. And then down here, this is kind of fun, poorly characterized, function unknown. So with Pseudomonas putida, 18.6% of their open reading frame, we have no clue yet what it does for the microbe. And so again, a lot of unknown, and that's the beauty of science. You get to constantly discover new things and apply that information. Okay, so this figure is messy, right? But in a beautiful way. So this is looking at the idea of, again, I think this is just, I can't remember which microbe they were studying, where they started to draw this map based on their studies. But you can see in the colored little rectangles, they're showing kind of the big picture idea of what these physiological pathways are doing for the microbe. And I think I have a better picture that shows how the cell membrane is such a workhorse for the bacteria. This really does not capture that. This picture is more focusing on the amazing active dynamic interior of a bacterial cell. How even though we can't see them with the naked eye, it is so incredibly busy what they're doing, and the amount of physiological reactions that they're doing at any 1 minute is astounding. And so even though they're tiny, they are mighty, and they have amazing capabilities, far more sophisticated in their physiological pathways than what we can do in our cells and tissues. And I hope to convince you of that a little bit as we work through this physiological pathways section. Oh, here's the other one. So this picture I like because now they're kind of zooming into the cell membrane, and they're trying to get you to appreciate how busy and dynamic the cell membrane is. So it's chock full of all kinds of proteins that allow for transport, but also are involved in key physiological pathways. And so, you know, the electron transport chain processes that bacteria do, they have to do that in their cell membrane because they don't have a chloroplast inside them. They don't have mitochondria, okay? So they have to put so much of that in their cell membrane. And then, of course, the artist is also drawing some of the things that might be happening. And really, this artist is focusing mostly on glycolysis, which if you took Gen Micro, you know that's a type of fermentation pathway. And so they're just kind of focusing on that. And then, oh, here's the TCA, the Krebs cycle, or tricarboxylic acid cycle. And then I don't know if they've mapped out, somewhere in here is the electron transport chain for maximum ATP production using oxygen. And I can't find it right now. It's there somewhere. All right, so I'm hoping you're starting to appreciate the complexity of these tiny organisms. They may be small, but they're doing a lot of things every single minute. Okay, this figure is nice because it's showing the idea of size distribution of the genomes. And then, so let's see, the open boxes are free-living microbes, all right? The gray boxes are obligate parasites. So the gray box is down here. The black boxes, little tiny ones here, and then a few big black boxes. Those are thermophiles. So if you are a thermophile, thermo means heat, phile means to love. And so that means you have adapted to high heat conditions. And then the boxes with horizontal lines are endosymbionts. And so again, you can see the shift. If you're a parasite or endosymbiont, you're going to be to the left of this chart because that's where you see the smaller genome sizes. However, the free-living microbes, on the other hand, you see some great diversity here. And that really does depend on what habitat they plucked these microbes from and looked at them. And so, again, thinking about your particular habitat, what ecosystem are you studying? The microbes in a free-living habitat like the soil system, for example. When you study those microbes, chances are many of those microbes, if not most, will have fairly large genome sizes because they have to have that versatility in on their chromosomes and plasmids so that, you know, as their habitat changes, and it can change quite quickly and it can change drastically, you need to have those genes, you know, available that you can turn on when things shift drastically, and you can turn those other genes on and start using them to adapt and survive. On the other hand, if you're an endosymbiont, you got your host to do most of everything for you. So your habitat doesn't change very much. You're in a very homeostatic habitat, and you can end up purging a lot of your DNA. Here is a nice comparison looking at, on the x-axis, the habitat variability. Oh, they spelled variability wrong. And genetic diversity on the y-axis. And you can see this positive correlation. The more diverse, more variable your habitat is as you go off to the right, the more diversity you get in your genetics, in your chromosome, your DNA, all right? So a very nice visual comparison here to show that the more variable your habitat, the greater your genetic diversity, right? This was looking at different forested ecosystems and swampland that they took this study from, all right? So again, looking at open reading frames and, you know, what is their genetic composition, it can vary tremendously depending on what habitat you pluck those microbes from, okay? So that's one of the big picture ideas I want you to think about. Okay, so we didn't even really get into any hard physiological pathways yet, but we will do that next time because I do need to give you at least some of the big picture ideas so you can get an appreciation for some of the major processes that might be occurring in different habitats. But I think we're out of time for now, for today, but does anyone have any questions on some of this that I have yet to answer? Okay, well, if not, then I will see the group 1 students tomorrow in lab. We'll do the next set of lab experiments, which is streak plate and spread plate, which are, there's PowerPoint slides posted in the EduCat. You could take a peek at those before we walk in to lab. That'd be great. And we'll look at the plates that we poured as well and see how those turned out. Okay, so everyone, I'll see some of you tomorrow, and everyone else have a good long weekend. Wow, it's not long. We're not at the holiday weekend yet, but we're getting there. All right, I'll see you later.
Yeah.